Dear friends, dear colleagues, good afternoon, good evening, and welcome to the EHAD webinar organized by the Gene Therapy Working Group, Pharmacy in Preparation for the Gene Therapy with Useful Information. My name is Ana Boban. I'm adult hematologist and head of Haemophilia Center in Zagreb, Croatia, currently serving as the vice president of EHAD. It is my great pleasure that the three ladies are here tonight with me. So we have two pharmacists from Berlin, Claudia Neuhaus and Katrin Ingram-Sieber, and we have hemophilia nurse from Madrid, Sara Garcia Barcenia. So it is my great pleasure because I think we are going to cover a very interesting topic of gene therapy, which is quite new to all of us. So we are going to really have some useful clinical everyday information on how to handle gene therapy in the pharmacy and how to handle gene therapy from the pharmacy to the patient, which will be explained by our pharmacists and nurses who really did this in their everyday clinical work. So please, your feedback is needed. Here is a barcode, so please scan it. And after the webinar, you can answer a couple of questions, complete the survey, and you will also receive a certificate of attendance. So with this, I would like to invite the first speakers, so Claudia Neuhaus and Katrin Ingram-Sieber, to give us the gene therapy preparation from pharmacist perspective. Please, if you have any questions, there is a Q&A code at the bottom of your screen. So please ask any questions and we will address it during the Q&A session after two of the presentations. So please, Claudia and Katrin, please, the stage is yours.
Thank you very much, Anna. So hello, everyone. And so both of us, Claudia and myself, we are pharmacists based in Berlin. And we are based in a community pharmacy. So maybe it's a bit special, the system in Germany, because we have very different sectors for inpatient and outpatients. And the gene therapy here in Germany was the two patients which we had were outpatients. So we had the chance to work together with our Haemophilia Center, who asked if we can support them in prepare the gene therapy. That's how we got here. So the general role of a pharmacist in gene therapy preparation is we process, of course, the prescription of the prescribing physician and the compounding and dispensing of the safe and secure medication. And it's a very individual therapy, of course, the preparing of the gene therapy. So we have to have the infusion at the right time and at the correct dosage for the patient. We supplied all devices used to inject the medication in very close exchange with the Haemophilia dosing center. So for this whole smooth workflow, the very strong collaboration with all the nurses and physicians and the GMP compliant compounder was very important in the whole process. To get an idea of the general workflow, so we have the Haemophilia dosing center, who of course selects the patient and gives us a prescription. We check the plausibility of the prescription, and if it's positive, then we order the gene therapy. If it isn't positive, then we of course have an exchange of information with the Haemophilia dosing center. So if it's ordered, then we have a GMP compliant compounder who prepares the gene therapy. And also there we have this very strong exchange because sometimes the information has to be really on the day of infusion about the health status of the patient. As soon as the gene therapy is prepared, it comes back to the pharmacy and we do the transportation and deliver it patient-specific on the day of application back to the Haemophilia dosing center. So we have a suitable patient in the dosing center. What happens next? We have the selection via the physician, of course. He evaluates individual patient data. For the plausibility check, the body weight is important at the day of infusion of the patient to get the right dosage. We have to be informed far along before the infusion of the day of infusion and how the blood analysis go. This is a very old school still, how it's often working in Germany, prescription, how a German prescription looks like. We are working on the digital version. So we get it in the pharmacy, do the plausibility check. If it's a no, we consult the dosing center. If it's a yes, we organize the infusion. And on the exact day of infusion, we consult about the health status. If the patient is in the dosing center and healthy and ready to go, we give the go to the GMP compounder. So how does the plausibility check work? We only have experience with Roctavian. We had 2 patients with Roctavian. There, the recommended dosage, as it's in the product information, it's 6 times 10 to the power of 13 vector genomes per kilogram. The dosage in the vials of Roctavian is 2 times 10 to the power of 13 vector genomes per milliliter. So we need per kilogram body weight 3 milliliter of Roctavian. And this is extremely important because the vials are deeply frozen with minus 60 degrees. So as soon as we start the process of thawing them, it has to be the right amount. So if we have an example of 70 kilograms body weight patient, we take it times 3. So we have the milliliters, so 210 milliliters of Roctavian solution, which we need for infusing. In one vial, which is produced, we have 8 milliliters. So then you had 26.25 vials. Of course, we then round it up to have the exact amount finally in the syringe for the patient. Another step for checking our own process is that we confirm how many syringes. So we use, or the Haemophilia dosing center used perfuser syringes, 50 milliliter perfuser syringes. So we checked how many syringes in the end should be in the transportation. For this example, it's 4 times 50 milliliters and 1 syringe with 10 milliliters. So if everything worked out with the plausibility check, we then give the order to the GMP compliant compounding. What is happening there? The incoming goods have to be inspected. The manufacturing has to be aseptic, of course. We have quality control throughout the whole manufacturing process. The batch documentation and batch release is important. And also the qualified packaging and transportation. So this is why we have this temperature logger at all times in the process, starting with when the product arrives in the GMP compliant compounder and the whole process of thawing the vials, transporting the vials until it's actually at the patient's place in the Haemophilia dosing center. There's just an example of a temperature logger, which we used. So what are handling aspects of gene therapy? Of course, it has to be aseptic handling and preparation in a clean room. It has to be clean room A/B conditions. By thawing, the vials have to be, so the thawing is at room temperature, and the vials have to be upright standing, no visible crystals, and no foam building, which means that you are allowed to turn every vial gently 5 times during the thawing. And during the thawing, it has to be UV protected. We had some dry runs, and we found out that if you have, for example, the 27 vials and you thaw them in one box, then the thawing time takes too long because they cool each other. So we then had to separate each vial for the thawing time to reduce the thawing time, because you have to be pretty quick because the patient is obviously waiting in the Haemophilia dosing center to get the time schedule done. The solution after thawing has to be clear and free of particles. As soon as this is not the case, you can't use it. For drawing up the solution, you have to use sharp needles, 18 or 21G, into the perfuser syringes as it was used in our hemophilia dosing center. The syringes are labeled, and then the packaging and transportation is prepared at maximum 25 degrees. So it depends on, it can be really hot here in summer, it can be very cold in winter. You have to be safe that it's neither too cold nor too hot during transportation. This is then how the packaging looks like. And again, the temperature logger is within the boxes. And you see that after the whole process, everything is documented with the temperature. As I said, we didn't try it out for the first time when the patient really needed the infusion. So we had some dry runs with our team to prepare them to be on time on the real day of infusion. So since it's very cost intensive, everyone was a bit nervous about the whole process. So we did some test runs, and also to find out what kind of infusion devices and in which quantities the dosing center wanted it. So in the end, they used the perfuser syringes to be safe. We used UV protection for the syringes as well. We had filters for each syringe. So when you take the next syringe, it's important that you change the filter, the infusion filter, and everything what the dosing center wanted and needed was delivered on the day. Transportation and delivery then looks like we have the GMP compliant compounder. It transfers the individual infusion really to a pharmacist. The pharmacist controls everything, the packaging, the temperature, the batch documentation, and actually the infusion then is transported with a pharmacist and GDP certified transportation to the hemophilia dosing center, where it's then delivered by a pharmacist to the nurse and the doctor who are in charge on that day for the patient. And actually the most important thing is the time management on infusion day. Since it's deeply frozen, so minus 60 degrees, and until the patient is cleared for infusion, then we can start. Then the thawing time starts, and it takes 2 to 3 hours under UV protection. And as I said, if you thaw them in one bulk, in one box, it will be prolonged, and you will have maybe problems with the time then afterwards, because within this 10 hours where the solution after being thawed, it's stable for 10 hours. And then the infusion has to be completed in this time period. So you have the quality control, you have the manufacturing, you have the packaging, you have the transportation, and you have the application, everything within this time period. This is why the exchange between all of the players is super important. And we were happy to have a really nice communication between the nurses and the doctors and the pharmacist. So the time of infusion depends on the dosage, of course, because then the infusion volume is different. And on the patient reaction, it takes between 2 to 5 hours, but I'm sure we'll hear far more information from Sarah afterwards in the next talk. And it can be, you can have infusion reactions, so also you should have a buffer of time to consider this problem that you need more time. So this is pretty much how we did our two preparations for gene therapy. That's our experience so far, and we are looking forward to hear what Sarah's perspective as a nurse is. Thank you.
Thank you very much for this very interesting overview. And now we are moving on to Sarah and her perspective as a hemophilia nurse. So how do you handle the drug outside the pharmacy from the nurse perspective? Sarah, please.
Thanks so much. Hi and good evening to everyone, and thanks very much to the organization for having me today. My name is Sara. I'm a study nurse working in Hospital La Paz in Madrid from 2014. And my topic is regarding the handling the drug outside the pharmacy from a nurse perspective. So I was thinking about that we nurses need to pay attention to in relation to gene therapy, and I detected like three big areas, beginning with the patient education, our role informing the patient and family about the process and the side effects that we are going to expect. Also, with the safe administration, because we as nurses must ensure that the process of administering gene therapy is performed correctly and in a safe way. And finally, regarding the monitor of effects, we have to pay attention to detect any immediately or long-term adverse reactions. So beginning with the patient education, in my point of view, I think it's very important to talk before the infusion with the patient and family and explain them that we are going to assume a lot of visits, especially in the first day and the first months, because we can detect this way the adverse events earlier. Also, that the post-infusion study follow-up is for several years, so we are going to monitor closely this patient during a lot of time. It's very important also to explain all the procedures, all the things that we are going to do at each visit when they come to the hospital, and also to check the knowledge regarding bleeding management, because probably we are going to find higher levels of thrombin generation. So the management of the bleeds must be, the patient needs to have this point super clear, and also what to expect on the day of infusion. Because the infusion day is a very important day where we have to explain all the procedures to be carried out. We are going to spend a lot of hours in the hospital. Maybe the patient is going to be admitted to the hospital, depending on the center of the protocols. And we are going to do a lot of procedures, different procedures. And also, it's important to be close to the cardiopulmonary resuscitation medicines and equipment. So continuing with the safe administration, when the gene therapy is prepared, we have the experience of clinical trials when the pharmacy call us, hey, it's prepared. So we in the clinical trials found that the medicine is prepared in a syringe. So as per protocol, it's mandatory to administer the 10% first and wait, check the patient, and then if everything is okay, continue with the other 90%. But that is the clinical trials experience. We have one approved therapy here in Spain, and it's prepared in 500 milliliters of sodium chloride. So when I was preparing this talk, I was like, I found this paper written by a group of pharmacists, and I think it's very cool. This was published in 2020, but they develop a table categorizing every adenovirus, every viral vector in a risk group. So the adeno-associated virus that is most commonly used in the gene therapy is level 1, and the author says that it's not necessarily associated, this kind of virus, with disease in healthy humans. And they provide us like these guides to manage the adenovirus. For example, the standard microbiological practice are followed. Work can be done in an open lab, bench or table, but should have doors separate from working space from the rest of the facility. So when the pharmacy call us and say, hey, you have the medicine prepared, we are going to find something like this, obviously dependent of each center, but we're going to find that we need to protect against light, maintaining a stable temperature according to the approved therapy here in Spain, between 15 and 25 degrees without PVCs. And we have to transport the drug in these conditions outside the pharmacy to the place of administration. In my center, we have the pharmacy and the Haemophilia treatment centers in different buildings, but in the same hospital. So we don't need to coordinate like a lot of staff, just to use the typical containers that we already have. And if we think in the administration, we already know that the medicine is stable 24 hours according to the license, but in a microbiological point of view, we already know that we have to administer immediately. This continuing with this paper, and because I think that is a really good paper, I found a lot of information there. This group of pharmacists recommend in-person handoff meetings between the dedicated pharmacy staff and the nurse who is going to administer the therapy. They recommend also to create committees to work safe handling the procedures involving the pharmacist and the physician, the infusion nurse, and the wait services, and also develop specific trainings or specific competences for each discipline: pharmacists, physicians, infusion nurses, investigators, or research personnel. And talking about the research, I'm a study nurse, so I very used to work with protocols. It's a cool way to work because the protocols help us to describe the objectives, design, methodology, especially the organizational aspects of clinical trials. But I think that this way of work increases the security to the administration process. And inside all the protocols, we are going to find always these flow charts. These are a very visual way to control all the procedures to be performed at every time point. So when I prepared this talk, I found this paper by Astermark and their colleagues that they develop a specific flowchart for gene therapy, thinking when we are in the transition between clinical trials and the approved gene therapy. So I think that is very, very useful to have this kind of tools when we are preparing our infrastructures to the arrival of gene therapy. And finally, talking about the monitor effects, we have the infusion day for nurses. Also, it's a special day. And in my opinion, I think that the things that we have to keep in mind is we have to ensure a quick access to medical support and medications. We have to be prepared in case of allergic reactions or any kind of reactions. It's okay if we review the patient notes, if we have all the knowledge that we can have of the patient that we have in front of. Also, cannot forget the 5 checks: correct patient, correct dose, correct medication, correct route, and correct time. And for sure, monitor post-infusion reactions. Looking again to the approved therapy here in Spain, these are the list of the monitoring effects. And I divide by color. Red color is the very frequent reactions that probably we are going to have one of these. In orange, less frequent, but already frequent. So I will pay attention to the last one, the infusion-related reaction, the immediately reaction that maybe we are going to have when we administer the gene therapy: hypersensitivity, infusion site reaction, dizziness, ocular pruritus, flushing, upper abdominal pain, urticaria, chest discomfort, or pyrexia. Also accompanied with a headache or flu-like syndrome, and we already know that this kind of therapy elevates the transaminases. Continuing with the monitoring effects, I think that it's important to take vital signs pre-infusion: heart rate, respiratory rate, temperature, and blood pressure. And I already said before, but I think it's very important to have quick access to direct access using a vein, have prepared adrenaline, oxygen, antihistamine, corticoids, saline solution, discuss what we are going to need with the hematologist, and it's very, very cool if you have a protocol before. Also, we are going to take the vital signs post-infusion, the same to detect changes in the patient. And if we look in another time points, we can see the short term. We are going to look for the effects derived from the corticotherapy or the factor levels. In a medium term, we can monitor or detect the alcohol consumption, the elevated transaminases. In a long term, we can focus in liver health or toxic habits. Everything, of course, depending and according to the protocols of each center. So to summary, my talk is, I think it's important to comply with clinical recommendations and strictly follow the administration procedures. already Catherine and Claudia says, that it's very important to have a clear and consistent communication between the healthcare team involving the patient and family. And as a nurse, I think also it's very important to remain educating on treatment advances and new therapeutic developments to be on date of that. And just to finalize my talk, I present to you the care roadmap that we make in Spain to try to implement the Haemophilia B gene therapy process in Spain. Beginning from diagnose, selection of the patient and treatment, informed consent, beginning of the treatment, short-term follow-up, long-term follow-up. But just to highlight that as nurses, the same that for hematologists, pharmacists, we are present in every step of this roadmap of patient that is going to administer gene therapy. So thanks so much for your attention, and waiting for your comments.
Thank you so much. So both from our nurse and for our pharmacist. So now we have quite time for discussion. So please, if you have any questions for both of our speakers, please just write it down in the Q&A session. So while the audience is thinking about the question, I have one for the pharmacist. So it doesn't look that complicated, at least from my side. I'm not that familiar with pharmacies, but do you have like an advanced preparation? This is the day for the gene therapy, so you clean, or the other schedule, so you're just focusing on the gene therapy, or it is just one in a row of preparation for the therapy of that day? What would you say?
No, you're absolutely right. The actual process is thawing something, getting it into the syringe, and then transport it to the correct patient at the correct time. So, but since it's more the time schedule has to be on time, so we would have in the pharmacy actually at least one, actually two persons completely focusing on the gene therapy, nothing else on that day of infusion, to get also the communication done, so to be accessible for all the players, because the nurses, I mean, it was nobody had it before, so everyone was a bit nervous.
Yeah,
sure.
I can completely understand that. And from my side, when I started, because in my center I wanted to start a clinical trial, so I got all these instructions, so how the pharmacy should look like, and then warned me, be aware that gene therapy is GMO product. I've never thought about it before, and they say, okay, you should have this really strict criteria for pharmacy to get approval from the ministry, because in Croatia we need Ministry of Health and Ministry of Science to get documents done, and you, the pharmacy, has the right accreditation for the preparation of the gene therapy. Is that the same in the other countries? Do you maybe know? Do you have the
Yeah,
same procedure?
yeah. At least in Spain, we need to have the approval of the ministry, and as we start before with the clinical trials, like this is a kind of preparation to the incoming of real gene therapy. So everything is set, and you have to do a lot of work, writing, provide the protocols to the ministry, as the same, or in Spain. And even in Spain, we have these advanced therapy committees, also included a CRISPR-Cas, and this kind of, we have these groups, working groups, national and also local working groups. So I think that there is a lot of people paying attention to this, and a lot of work are being done. Like, for example, webinars like this. So I think it's cool.
Yeah. So in my experiences, when I talk to my pharmacy, they say, okay, we have all the requirements that is needed, but actually to get the approval to fill in all the documents, it took a couple of months actually to get all the documentations in order to start the treatment. So I have one question from the Q&A. Which compounder do you use? I think it's for Catherine and Claudia, the question.
So we use the Medios Manufaktur, it's called. It's based in, because you need, it has to be very close transportation ways. So we are working or collaborate very strongly with Medios Manufacture.
Okay,
Because
thank
obviously
you.
that's the only thing you really need, a GMP confirmed manufacturer, and also with the clean room, this won't be possible for every pharmacy. Yeah.
Okay, thank you. And we have a question for Sarah. With these therapies, is there any risk of viral shedding by the patient? Are they isolated away from other patients?
Well, no, they are not isolated away from other patients. In my experience with clinical trials, we measure a lot of weeks before. I don't remember how many weeks, but already studied. I don't have this in the data in mind, but the pharmaceutical companies knows the viral shed in blood, urine. Also stool and semen, I think. So they study all the biological human fluids, and they have the data, but I don't know. Sorry, but pharmaceutical companies have. I'm not sure. I cannot say to you completely the hours or the sets, the viral seeds, but I know that the pharmaceutical companies know, and we extract a lot of samples, biological samples, in order to see when the virus is now cleared from patient.
Yeah. But so to be practical, if the patient is staying overnight in the hospital after the gene therapy, would it be alone in his room with his own bathroom, so not to get in contact with other patients?
Yeah, yes. We sometimes we usually do that, but sometimes the hospital has a high overload, and sometimes we cannot avoid that. When it's possible, I think it's preferably to do that.
Okay.
So this is maybe interesting because this is a difference in Germany. So the patients actually weren't overnight in the hospital. So they got the infusion, stayed there for a few hours, and then were close to the hospital. So if they are not living in Berlin, then they weren't allowed to go home like 100 kilometers, but they didn't stay in the hospital.
I think that could differ from sites, countries, protocols. There is a lot of different ways to do.
Okay. So I have another question for the pharmacist. Say you indicated it took quite a long time for the medicine to thaw. What was the volume of the medicine in the vial?
Per vial, it's 8 milliliters. So, and I mean, long, but you want to have it as short as possible, as close to 2 hours as possible. And we had this problem if you have the vials close together, then they cool each other. So it's better to have different boxes per vial because it has to be UV protected. So then the thawing is much quicker.
Okay. And we have another question, I think for both of you. So do you have any paperwork, SOPs, both from nurses and for pharmacists that can use? Do you have a written document how to use it?
We are prepared. I'm sorry, Catherine.
No,
Go
go ahead.
ahead.
Go ahead.
You. We are starting to write in protocols and prepare our installations, because in my personal point of view, I see difference when we are in the clinical trials and when now the therapy is outside the clinical trials. So we are working on that, but they are under development until now.
Okay. For the
Yeah, so
pharmacy?
we actually do have an SOE. We have one, yeah, written down.
So we have another question. So you said you monitor the temperature of the vials and syringes from thaw to administration. How do you do this? Do you confirm the temperature has been maintained before administration? So I would
So
say yes. So you show these little thermometers. So are
yes, so
there?
you would have these thermometers at all times, and then as soon as the package arrives at the hemophilia dosing center, then the room temperature should be standardized in the room where the patient is. So as soon as it's out of the packaging, then the logger is out of the story. Yeah. But then you would have an environment in the clinic with room temperature.
Yeah. Okay. So I have another question for you, for the pharmacists. So do we have any procedure for disposing of the material that was in the contact with the gene therapy? And same for the nurses. So it just infect, you have this infected disposal, package, or it's some special procedure? How do you handle this material? Or you just throw it in a like infection?
So I only can, because I'm not 100% sure. I'm sure there is a special disposal in the so Medios manufacture, because they have it written down, everything of the whole process. I can't actually answer you how exactly the disposal. If this is of interest, I can give it in later.
Okay, thank you.
When we administer the therapy, like the infusion bag or the syringe, we use the same disposal as we use as chemotherapy. I'm sorry, chemotherapy. Sorry. So they are standardized products, and then they dispose and burn, and they have their own shops.
Okay, thank you. So it seems that nurses have really a lot of work around the patient. So is one nurse enough for handling the patient, or there are two nurses or more nurses with the patient at the time of gene therapy administration?
I think this will be depend of the nurses that you have available to do that. I think that, but in my experience, one nurse is enough. But we are not alone. We have a lot of colleagues around and in the clinical setting. So I'm not alone. If I need something, I will have colleagues close to me. So that makes me feel like calm. But I think that one nurse can, I mean, you have specialized nurse working with these newborns with a lot of complex therapy. So I think that one nurse can be enough, and she are not going to be alone. So
yeah, and I think if you're a patient receiving gene therapy, it can be quite emotional, so probably a bit excited. So I think from my point of view, it will be good that nurse already knows the patient from the beginning, so they have like better relationship, that so the patient can share some problems.
yeah.
So what do you think? So with this hub and spoke model, so patients sometimes will travel from one center to another, and for the first time meeting a nurse, I mean, how would you deal with it? Because I think it will be much easier if you're familiar with the patient and he's familiar with the nurse that is taking care.
Yeah, full agree with you. I don't have that problem because my center is like the hub and spoke, but are they both? But if I try, I think that I will feel better if it's my nurse and my hematologist and my staff, and I'm really curious how things are doing in other models, like if you have to go outside to another center and to be treated by another professionals. I think that maybe this could be a challenge, but I don't see like no, I'm not going to put gene therapy because that, no, it's a challenge. We need to prepare, we need to think. Now you make me think that I am going to be the new nurse from a lot of patients, so we have to think about that. Yeah.
And I have another question. So we know that it's really important that patient get whole the dosage of the drug, because if you stop prematurely, doesn't receive, obviously he won't have the good response in the increase of factor VIII or NAL levels. And then there's no possibility to redose him later. So regarding this immediate reactions, I think you should be very careful when to decide, okay, this is the reaction when we won't give the treatment anymore, and this is okay, we can slow the infusion or give some premedication. So I guess it has to be really quick discussion between nurse and the physician that is treating patient. What do you think about? What's your perspective on that?
When I have administered gene therapy, the hematologists are very close to me. So I think obviously it's very important to have everything prepared in a protocol or something and discuss with the hematologist. But I think that I don't see problems like that. If we are like close and we have everything prepared, I think that things are going to be smooth without complications. And if the patient experience a big reaction, we are going to stop and medicate and follow the hematologist's instructions. So I'm not sure how to answer the question in a clinical point of view, but I think that the most important thing to protect us is just to act a team.
Thank you. So it seems that there are really quite a bit of different details you need to be aware when starting the gene therapy, and you already mentioned some specific training for nurses. So, in your idea, how would you organize? I mean, it's not easy because the hemophilia centers are not that many, so each country one or two, and we don't have much experience in the gene therapy. So, how would you propose, or what would be your vision on how to educate other nurses from other centers? So, apart from webinars like this, how would you?
It's a good question. Maybe the academic societies can develop this kind of content, or me as nurse, when make the clinical trials, I don't have any training more than the training provided by the sponsors and the pharmaceuticals companies. So my training was like the performing of the clinical trials. I think that maybe webinars like this or the academic societies, or maybe we as nurses must to say, okay, I'm not so familiarized with that, so I need to look for information because I need to be prepared. Because when you administer the gene therapy, for me it's very important to be prepared, to know, okay, this is a reaction, now I know how to do this, and have everything prepared to save to make safer the process of administering gene therapy for the patient, but also for ourselves.
Yeah. And for you both, do you see there is a big difference between preparing gene therapy in clinical trials and in real life outside of clinical trials, or is more or less the same procedure?
I mean, we pretty much only have experience without out of the trials, so we only
Okay.
have the real
Okay.
life experience. We haven't been involved in clinical trials.
Okay.
I think that the clinical trial is like pre-training. So maybe when I think clinical trial can be maybe more exigent or more like hard to follow all the procedures, and when it's lablet, maybe we can relax at some point regarding the timelines or data or this stuff, but more or less are the same.
Okay. Thank you. So from your side, from your clinical experience, it did everything went well with the infusion? Were there any reactions, allergic or some? No. So how long does it? How long the infusion lasts? So you start first 10% and then a little bit slower, then longer. But taking in total, how long does it take to infuse the drug?
It depends if there are reactions or not. I'm as I mentioned, the syringe, when we administered the first 10% and then the other was in clinical trials, not in the approved therapy. But I cannot remember, but not so long. Maybe in 1 hour in total or something like that. Not so long. When I was like in the clinical trial, the first time that I'm going to administer gene therapy, like wow, and I was like, I saw the first time the syringe, I was like a bit disappointed because sounds like gene therapy, and at the end it's like big volume, yeah, but it's like a flush when to treat factors, so it's not so complex. It's everything that we do all time, just to administer in a pump, in a syringe.
Okay, so one morning in a clinic, and you can apply gene therapy if everything went well.
Yeah.
So, yeah, with this, I would like to thank you all for really nice discussion, for very nice and open and honest answers. And I think this topic again emphasized the need for multidisciplinary team in the management of patients with hemophilia. And we have now pharmacists, which have been with us for a long time, but now they actually have a new, very, very important role. So, for all our listeners, please visit. You can have link on this. You can scan this QR code, and it will take you to EHA gene therapy trial database. And please check what is new, what new data has been added, because we update this database quite commonly. And please, in the end, do not forget do not forget to answer this really short questionnaire, because your feedback is very important to us in tailoring our future webinars. So with this, I would like to thank again all the speakers, all the participants, and have a nice evening. Bye bye.